Hello, everyone. Good morning.
Morning.
Good to see you. Happy Friday. Yes, if you wouldn't mind, go ahead and type your first name or your nickname into the chat for me so I can take attendance. Thank you. Very good. All right. So today we're going to be finishing up our week-long discussion of carbohydrates and carbohydrate catabolism. The last time we were together, we started into this next exercise about fermentation. So that's where we'll jump in today. Today we're going to be looking at ways that we can learn whether or not microbes are capable of fermenting various sugars, various carbohydrates. Remember, we said when microbes ferment, they produce certain products, and those products include acids and alcohols and gases. So we're going to be looking at testing using those products, because we know that if we can figure out what a microbe is capable of doing, we can sometimes use that information to identify that microbe. And that's often our purpose in a microbiology lab, identifying unknown species of microbes. So before we jump back into the fermentation slides, does anybody have any questions from our last time together, or any of the lecture material that you've been viewing, or any of the material you might be reading in your textbook? Any questions about anything you've been working on this week? Feel free to type your questions into chat if you do. I'm just going to pull up our Blackboard page and our to-do list for next week, just so we can look ahead at what's coming up, just so we can prepare for next week. So that's what you should be seeing right now on your screen. This is the to-do list for the week of March 29th, which is next week. We're rapidly coming to the end of March. And next week, the lecture topics are going to be all about genetics, or essentially what's happening with the DNA molecule inside of microbial cells. So the first part of this topic is this process called DNA replication, and another very natural process called mutation, and how those processes occur. And then the second half of the subject involves how DNA is used inside cells to build proteins. So it involves the process of transcription. And that's the process where the DNA molecule is used to make an mRNA. And then translation takes the mRNA molecule and uses it to build protein. So two very different processes: replication and what we call gene expression, transcription and translation. And those will be coming up next week. In laboratory next week, we'll be talking about protein catabolism. So we'll move away from carbohydrates next week, and we'll move into proteins. So before I jump in, I did want to make one, I guess, public service announcement. If you didn't catch the news conference yesterday, or you didn't see the news yesterday, our governor has accelerated the vaccination scheduling for New Hampshire, officially as of yesterday. Originally, the plan was that people your age would be available to get vaccinated starting May 1st. I don't know if you had heard that in the news, but as of yesterday, that date has been moved up. So it's going to be in April instead. I want to say it's April 1st, but I think it's a little bit later. Hold on just a sec. I was close. It's April 2nd. April 2nd will now be the day that you can go on to the newhampshire.org website, to the vaccination page, and you can schedule yourself an appointment to get a COVID vaccine. So that's good news. That just means that more people will be eligible to be vaccinated, and that's always good news. All right. Well, if there are no questions for us to talk about, I'm going to go ahead and jump back into our fermentation slides around the place where we were last time. So remember, we're going to look at 3 tests today. We're going to look at what's called the Carbohydrate Fermentation Test. It's a great name because it tells us exactly what it's looking at. We're going to look at a test called Mrvp, and we're going to look at the Citrate Fermentation Test. The first one is the Carbohydrate Fermentation Test. And remember, this test allows us to test the ability of microbes to ferment specific carbohydrates. In other words, we're looking to see if they have an enzyme that's necessary to take something like a disaccharide and break it down to get glucose that they can ferment. Remember, glucose is sort of the key monosaccharide that cells use to make energy. And of course, by energy, I mean ATP. Sometimes microbes have the ability to take a slightly more complex carbohydrate, like a disaccharide, and get the glucose out of it, essentially, by breaking it down. And then they can ferment the glucose. Remember, too, that fermentation is a much less efficient way to make ATP. It's made through what we call substrate level phosphorylation, which just isn't as efficient as the way it's made during cellular respiration. During cellular respiration, it's made through oxidative phosphorylation. And during fermentation, it's made through substrate level phosphorylation. So we take a tube of the carbohydrate fermentation medium, and that medium, remember, starts off with a red color. We're also going to put a Durham tube, a little empty glass test tube, upside down into the medium, because that's going to help us know if the microbe can produce gas when it's fermenting. And we're going to put the inoculated tube into the incubator and let the microbe grow. We're looking for 2 things in this test. We're looking for a color change to yellow. If we see yellow the next day, it tells us that that organism fermented the sugar that was in the tube and produced an acid byproduct. And we're going to look for gas. We're going to look for a bubble of gas. We're going to look for the liquid in the Durham tube to be displaced by a bubble of gas. Now, on this slide, in this picture, you can see 3 tubes. You can see an uninoculated control tube, and then you can see 2 tubes that have been inoculated and incubated with 2 different organisms. And there's some results typed over the tubes. This first tube on the left has turned yellow, so that's a positive result for acid production, and that's why you've got an A there. Carbohydrate fermentation tests are usually reported as A or A/G. So the A means that only acid was produced. And the G means that acid was produced, the tube turned yellow, and some gas was produced during the process of fermentation. I think if you look, as I enlarge the image, you can see at the top of the Durham tube, it's now clear. Over here, it had the yellow liquid all the way up to the top of the tube. And in here, the yellow liquid has been displaced. It's been moved down because there's a big gas bubble right here. Remember, there are two common gases that are produced during fermentation. One of them is carbon dioxide, and the other one is hydrogen gas. It depends on the pathway. It depends on the pathway. And of course, these gases can also be converted into acids. So you're always going to have acid if fermentation occurs. The question is, are you also getting a gas produced? And that's what this test allows us to see. So on this slide, what you're looking at is some homemade carbohydrate fermentation medium. It is perfectly fine to make your own in a laboratory. You can also purchase the carbohydrate fermentation medium. That's what these tubes are. This is a commercially available version of the test. But, you know, in some laboratories, it makes more sense financially to just make your own medium right in the lab. So this is the uninoculated control tube, and you can see the Durham tube sticking up. And then we've got 3 different organisms in these other 3 tubes. Now, part of the reason I chose this picture to show you is because I want you to notice how different the control tube looks, the uninoculated tube looks, compared to this picture. This is commercially made medium, and this is the medium that was made in this laboratory by hand, sort of custom made. A lot of times when people choose to make their own carbohydrate fermentation medium, it's because they want to add something into it that they can't get commercially. And when you do that, when you change the ingredients in the medium, sometimes the starting color of the phenol red changes. So in a very classic carbohydrate fermentation medium like this is the commercially available medium. The phenol red is very red. It makes the medium very red. But as you can see, that's not necessarily where you will begin color-wise if you start making changes to the medium. You can see that it's really important to run an uninoculated control tube when you're using medium, especially when you're using custom-made medium, because you're going to need this tube for comparison. These 3 tubes were inoculated, this one with E. coli, this one with Shigella, this one with Pseudomonas. And as you can see, 24 hours later, the Pseudomonas tube is the same color as the uninoculated medium, the control medium. So that tells us that this is a negative result. You can imagine that if you hadn't put this tube in the incubator, if you had left the control tube out and just run your tests, somebody might look at this tube and think, oh, well, that's not red. I mean, it's not yellow either, but it's not red. No, it's not red, but it's the same color as the uninoculated medium, as the medium at the beginning. So there has been no pH change in that Pseudomonas tube, and that tells us that fermentation did not occur in that tube. So it's really important to take a tube of your carbohydrate fermentation medium that you did not inoculate and put it in the incubator right along with your other tubes as your control tube. You want that uninoculated tube to be handled the same way that all your other tubes get handled. The only thing that differs is that it doesn't have any cells in it. So put it in the incubator, let it incubate, just like these tubes will incubate. and use it as your comparison the next day. If your experimental tubes are the same color, then nothing happened, no fermentation. If your experimental tubes are a different color, and for this indicator dye, it's going to be yellow, then you know an acid was produced. The pH in these tubes is very low, it's acidic, and that's what caused the color change. Remember, don't forget the Durham tubes. So yes, these are positive for acid production, but you also have to look at the Durham tubes. And I know it's hard to see because of the color, but this tube, this Durham tube, is still full of that yellow medium. There's no gas in this tube. Now look at the E. coli tube. The yellow medium has been displaced. It's been pushed downwards because gas has accumulated in the top of that tube. So this tube would be reported as acid, or capital A. And this tube would be reported as A/G, acid and gas. All right. Does anybody have any questions about how these tubes work? About why we run them? About how we use them? Remember, this is a broth. This is a broth in a tube, very liquidy. So when you get a color change, you're likely to see it all the way through the tube. You're going to read it at approximately 24 hours, as long as the organism is growing. You're looking for very particular types of sugars, the ability of the microbes to ferment those particular types of sugars. Remember, they are using the glucose to make the pyruvate, to make the acid or alcohol or gas. But some microbes can get the glucose from a more complex sugar. So some microbes, when we say a microbe can ferment lactose, what we're really saying is that microbe can take lactose and with an enzyme, it can break it apart into glucose and galactose, because those are the two sugars that make up lactose. So yes, it's fermenting lactose by breaking it apart, getting to the glucose, converting the glucose into pyruvate through glycolysis, and then taking that pyruvate and making either acid, alcohol, or gas. Lots of microbes can ferment several different sugars. They have a nice assortment of enzymes to break down these disaccharides and get to the glucose. All right. So I don't have actual pictures for you today of our results. They didn't come out very well. So instead, I pulled some images out of a textbook that will demonstrate the results. Remember, we're using 5 different organisms today: E. coli, Enterobacter Erogenese, and Klebsiella pneumonia. Those are our 3 gram-negative coliforms. They are typically known as lactose fermenters. Then we have Salmonella, or also called Shigella sonnei, and Pseudomonas aeruginosa. Those are also gram-negative, also intestinal, but not coliforms. So we have 5 organisms to take a look at today. And we're testing for glucose, lactose, and sucrose. Now, I just told you that when we say something ferments lactose or ferments sucrose, what they're really using is the glucose that they're getting out of the lactose, that they're getting out of the sucrose. So why look at glucose? This is almost like a double check. We're going to double check that they are capable of fermenting the glucose. Remember, we also saw the OF test this week: oxidative, fermentative. And that test has glucose in it as well. But remember, the OF test is usually used to determine if a microbe can ferment. If it can ferment, again, glucose, this very common, universally used carbohydrate. The carbohydrate fermentation then takes the organism and says, what can it ferment specifically? What sugars can it break down to get to glucose? Some people don't want to run both tests. It's inefficient. It takes too much time. So they will just run the carbohydrate test, the carbohydrate fermentation test. And if they do that, they add in the glucose tube first, just to double check that the organism can ferment glucose. It's easy. Just set up one tube for glucose, one tube for another sugar, one tube for a third sugar, a fourth or a fifth or a sixth. You can run as many of these as you want. But sometimes you have skipped over your OF test. You don't yet know if the organism is even capable of fermenting. So you add the glucose tube in as well. That's going to tell you if the organism is capable of fermenting. So here's our first comparison. This is between E. coli and Klebsiella. Remember, I said there are 3 gram-negative coliforms that we're looking at today: E. coli, Enterobacter, and Klebsiella. I'm leaving the Enterobacter out for right now, and there's a reason for that. I'll get to the Enterobacter, I promise. But for right now, we're just going to compare 2 of them, the E. coli and the Klebsiella. We're going to look at those 3 sugars in 3 separate tubes for each organism. So even though I'm only showing one set of tubes here, I ran one set of tubes for each organism. Now, over here, we have the control. So this is what the tube looked like before I put the cells in. You can't see the Durham tube very well, but it's in there. It's drawn in there. It's full of red liquid. So glucose tube, 24 hours after incubation. So I put cells into the medium. I put the tube into the incubator. 24 hours later, I pulled out the glucose tube and it looked like this. I pulled out the lactose tube, it looked like this. I pulled out the sucrose tube, it looked like this. And these were the results for both organisms. Both organisms had an identical result for this test. That's what is shown down at the very bottom of this picture. Again, very hard to read because it's small. E. coli for glucose is A/G. Acid, because the media turned yellow. And gas, because there's a little bubble at the top of the Durham tube. In the lactose tube, same thing. In the sucrose tube, same thing. Yellow color, gas bubble. So AG all the way across. Now think about what this means. E. coli is able to ferment glucose. It's able to take the glucose molecule and, using the process of glycolysis, convert the glucose to pyruvate. Then the pyruvate gets converted into product molecules, including acid and gas. E. coli also has the enzyme necessary to break lactose down to get glucose and then ferment it, turn it into pyruvate, and then into acid and gas. It also has the enzymes needed to break down sucrose, get the glucose out, and ferment it. Turn it into glucose, which then gets converted to pyruvate, and then into acid and gas. Very, very adaptive organism, E. coli. And it turns out Klebsiella, in this case, Klebsiella pneumonia, has the same abilities. Acid and gas for all three carbohydrates. Now, is that clear to everybody? Is it clear how you would interpret that test? You should know how, if I told you, for example, on a quiz or an exam, that you inoculated one of these tubes and it had, say, fructose in it, or it had mannitol in it, or it had maltose in it, and I told you that you inoculated it with an unknown organism, and you took it out the next day, and I told you what color the tube was, and I told you whether or not there was a gas bubble in it, you should be able to then explain to me, is that a positive or a negative test? And what does it mean? What have I just learned about that organism? Okay, so if it's not clear, let me know. Type it into chat if you want me to go through anything else. Okay, otherwise we'll go on and we'll look at some more results. Okay. So now take a look at these tubes. I ran a second set of tubes for this organism called Shigella sonii. Now this is also an intestinal organism, a fecal organism. It's also a gram-negative rod, but this organism is not a coliform. And what that means is it cannot ferment lactose. It doesn't have the enzyme that's necessary to break lactose down into the two sugars that are in lactose, glucose and galactose. That's all lactose is. Two simple sugars, glucose and galactose, that have covalently bonded together. And remember, lactose is found in milk. This is what we call milk sugar. So the organism was tested for glucose, it was tested for lactose and sucrose, just like the other two organisms. I inoculated the media. I put the 3 tubes or the 4 tubes into the incubator. 24 hours later, these were the results. So here's the tube that has glucose in it. This one turned yellow, and it has a bubble at the top of the Durham tube. So Shigella has the ability to ferment glucose. It can take that glucose and it can convert it to pyruvate through glycolysis, and then take the pyruvate and turn it into an acid molecule and a gas. So it gets an A/G. Now, what about lactose and sucrose? Well, these tubes are still red, and there's no gas in the Durham tube. This is a negative result for carbohydrate fermentation. This is an organism that's capable of fermenting. It's capable of fermenting glucose, but it doesn't have the ability to get glucose out of lactose. It doesn't have the ability to get glucose out of sucrose. Sucrose is just a disaccharide. And remember, sucrose is table sugar. It's a disaccharide that has glucose in it and fructose. Those two are bonded together covalently, and they create table sugar. So what Shigella does not have is an enzyme that can break that bond and release the glucose so it can be fermented. If it had the enzyme, it would be able to take the glucose and ferment it, just like it did in that first tube. So we know by looking at that lactose tube that it doesn't have the enzyme, because that tube stayed red. Same thing with the sucrose tube. So unable to ferment lactose, unable to ferment sucrose. Now take a look at Pseudomonas. This is our other gram-negative rod gastrointestinal organism, not a coliform. It looks very similar to the Shigella, doesn't it? Yellow tube for glucose, red tubes for lactose and sucrose. But there's an important difference, and that's right here. Remember, when Shigella ferments glucose, it produces a gas as well as an acid. When Pseudomonas ferments glucose, no gas. All right, that's an important distinction. They're both fermenting, they're fermenting glucose, but they're using a different pathway to do it. Remember, there are lots of fermentation pathways. There are lots of ways that microbes can take a glucose molecule and make products with it. Different pathways. And you're always going to get some combination of acid, alcohol, gas produced, but it's done through different pathways. So obviously, Pseudomonas is using a different pathway because it's not getting the exact same products. So that's an important thing to know about that organism. So if I were to put all of these results in a table, it would look like this. We had 3 organisms that are able to ferment glucose: E. coli, Klebsiella, and the Shigella. But, 4 organisms and Pseudomonas. But only 3 of them were able to produce gas as one of the products. So all of them are fermenters. They're glucose fermenters, but they vary in their ability to ferment other sugars. The E. coli and the Klebsiella could ferment everything that we threw at it, but the Shigella and the Pseudomonas, only the glucose. And if you think about it, it's an interesting finding because these 2 organisms are coliforms, and these 2 organisms are not coliforms. And so when we designate organisms like that in medicine, there's physiology behind it. The fact that some microbes are able to ferment lactose is important because that's not only is that a characteristic that that organism has, but it tells you a little something about how it lives. So in your intestine, when you drink milk, and that milk comes down into your intestine, there are going to be organisms that are capable of fermenting it. When you eat sugar and that sugar gets into your intestine, there are going to be organisms that are capable of fermenting that sucrose. Other organisms can't do it. So these are things that, that are clinically important as well as just interesting scientifically. They help us identify organisms, and they also help us understand why certain organisms cause certain things to happen in the human body. Okay. So I want to move away from the carbohydrate fermentation test now and move into the second test on our list today, which is this methyl red Vogues Proskauer test. Just to review, these are 2 separate tests, but we run them together because they are related to each other in terms of the information we can get from them. These 2 tests are going to give us an indication of what kind of acid, generally speaking, is being produced through fermentation, and what's happening to that acid. Does the cell do anything with it in particular related to converting it into something neutral? And one of the most common neutral products that get made from acid is this molecule called acetoin. So again, the Mrvp test is kind of stepping from the carbohydrate fermentation test a little bit further. If I know that an organism can ferment glucose, or lactose or sucrose or any sugar. It's interesting now to know, okay, so an acid's being produced, but tell me something about that acid. Is it a strong acid? Is it a weak acid? Is the cell doing something immediately with that acid? Because there are certain fermentation pathways where the acid product is almost immediately converted into a neutral product called acetoin. So those are the things I can learn from this test. So 2 tests, 2 different media, but they're related to each other. The MR test will tell me the pH level of the acid product. I'll be able to know if it's a stronger acid with a pH lower than 4.4. If it's a weaker acid with a pH between 4.4 and 6, or if it's a weak acid with a pH of about 6 or more. That's the MR test. And the VP test will tell me, is the organism then taking that acid and turning it into a neutral product called acetoin? The MR test is easy to run. You just inoculate the media, put it in the incubator, read the results. The only tricky thing with the MR test is that it has a different indicator dye in it than we're used to. We're used to seeing media start red and turn yellow. And this media does exactly the opposite. This media starts yellow and turns red, some shade of red, depending on the acid that is being produced, how acidic it is. If it's very acidic, the media turns very cherry red. If it's less acidic, it starts to become more orange. So the hardest thing to know about the MR test is that you got to remember that the colors are exactly opposite to what we're used to seeing. The media begins yellow. And when the acid is produced, it changes it to some version of red or orange. The VP test is, remember, a little more complex because I'm going to put cells into the media. I'm going to inoculate the media. But when I come back the next day, I'm not going to see anything different. I have to put a few drops of a reagent chemical into the tube and then read it. So you can't forget that reagent. You'll think you have a negative result for every VP, but you're not going to see results until you put that reagent chemical in. And what we're looking for is a red ring to form, generally at the upper part of that culture tube. That's a positive test. That's telling us that the organism fermented, made an acid, and then converted the acid into acetoin, a neutral product with a pH of 7. Okay. So here are the 2 tests side by side, because again, we tend to run these together. MR test, here's the uninoculated media. It has a dye in it called methyl red. It starts yellow. A positive result is going to be some kind of a red to orange color. The more red it is, the stronger the acid. The less red, more orange it is, the weaker the acid. Pretty simple test all in all, as long as you remember how to read it. Here's the VP test. This is the medium that starts off just a really ugly looking color. It's sort of a beige green. It's just an unpleasant looking media. We're going to inoculate it. We're going to incubate it. We're going to take it out of the incubator and we're going to put a few drops of reagent into it. And we're going to look for this. If you see a red ring forming towards the top of the tube, you've got a positive Vogues Proskauer or a positive VP test. And the way you interpret that is that the organism has fermented the sugar and has then converted it into acetylene. If you don't get a red ring, it just means that there's no acetylene in the tube. So that last step did not happen. Both of these media use glucose as the carbohydrate. All right. I said a few minutes ago that I was holding off on looking at our fifth organism. Remember, our fifth organism today is Enterobacter Erogenese. This is another gram-negative fecal coliform. And we know that Enterobacter and E. coli are both gram-negative rods, both intestinal or fecal organisms, both coliforms. That means they're both able to ferment lactose. So you can imagine that sometimes it would be hard to tell them apart. If I ran an OF test, it would be the same for both of them. If I ran a carbohydrate fermentation test, it would be the same for both of them. So how in the world do you tell them apart? Well, it turns out for these two organisms, one of the ways we can tell them apart is with the Mrvp test. So what you're looking at over here on the left are the MR tubes, and over here are the VP tubes. Now it's a little confusing in the picture, so let me explain. These are the tubes that came out of the incubator after incubation and after the reagent was added. So Enterobacter Erogenese, E. coli, after 24 hours of incubation, methyl red. This is the Enterobacter Erogenese test for the VP. After 24 hours of incubation, after the reagent was added, here are the results. And here is E. coli. So the interesting thing about these two organisms is they essentially give opposite results on the Mrvp test. Remember, if you see yellow in the MR, it's a negative test. So Enterobacter Erogenese has a negative MR test. E. coli has a positive ER test. It has produced a strong acid. Over on the VP, it's the opposite. Enterobacter has the positive, and E. coli has the negative. So for these two organisms, you get one of each result, and they're opposite from test to test. E. coli is positive MR, negative VP. Enterobacter is negative MR, positive VP. So you can see that if you had a situation clinically where you were trying to figure out which one of those two organisms was in a patient's sample or in an environmental sample, this is a test you could use to distinguish them. Okay, the last test we're going to look at today is this citrate fermentation test. The technical name for this is the Simmons Citrate Test, but everybody just calls it the citrate test. So you are welcome to call it the citrate test for me. Simmons is really the name that got associated with the agar, that the particular recipe for the media, and this is somebody's name. So remember, this test is looking for the ability of an organism to ferment this molecule called citrate. And what makes this test unusual is that citrate is not a carbohydrate. When we think about fermentation, we think about carbohydrate. We think, oh, sugar, you're fermenting a sugar to make acid alcohol gas. Some organisms have the ability to ferment this molecule called citrate. So it's a totally different pathway. Citrate is a molecule that shows up in biochemistry in several different metabolic pathways, even in our bodies. For example, citrate is one of the intermediate chemicals that we find in the Krebs cycle. So citrate is around in a living cell because it gets produced in a couple of different metabolic pathways. Some microbes, some bacteria have the ability to take that citrate molecule out of whatever pathway it's in and instead ferment it. Why would they do that? Well, remember why organisms ferment. Remember why the organism ferments. It needs to make ATP for some reason. It's making ATP. So sometimes under certain circumstances, let's say there's not enough glucose around, maybe it prefers to ferment glucose. If there's citrate, it'll ferment that instead and get its ATP. So it's an interesting trait for an organism to have, the ability to ferment citrate, and it's a very easy test. Citrate agar, or Simmons citrate agar, you can buy commercially or you can make it. A whole lot of people just make it because it's easy to make and it's inexpensive. And the nice thing about making it yourself is you can decide what kind of container you want to make. You can decide whether you want to just make a slant tube. That's very easy. Or you can pour the agar into a petri dish and make a plate. Or you can make it as a broth. Very easy. So all we're looking for with the citrate agar is a color change. And in this case, we're starting off with a dark green color, and we're looking for a color change to bright blue. So this is what the citrate agar looks like when it's uninoculated. If the organism that we spread down this slant, if it starts to ferment the citrate in the agar, it will turn this lovely blue color. Remember, slants give us this nice slanted diagonal surface for when we don't need individual colonies to grow. So we don't need a whole lot of surface area. We can take our cells and we can just sort of smear them down that slanted agar. And it's a common form of agar to use when you're doing biochemical testing like this. The plates, you can also use the plates. It's just that typically when we use plates, it's because we need all that surface area because we're trying to spread the cells out enough that we can get individual colonies to grow. So this would be a negative result for citrate fermentation. This would be a positive result. I don't actually have inoculated positive tubes for the citrate slant agar, but I do have an uninoculated tube here. So you can see the slanted agar, and it's an agar, it's solid, yeah? And it's got this really deep green color. So if I were going to inoculate this, all I would need to do using my best aseptic technique is keep the tube on a slant, remember? When I take the cap off, I want to keep it in my hand. I want to sterilize my inoculating loop. I want to dip it down into my culture aseptically, and then that's going to go into the tube, and I'm just going to smear the agar with it. You start down at the bottom and you smear your way out of the tube. Remember, you don't want to touch the mouth of a test tube like this any more than you absolutely have to, because you might introduce a contaminant. And as soon as your tube is inoculated, into the incubator. So I don't have a positive slant tube to show you, but I do have a positive broth tube I can show you. And that's this one. Look at that color change. It's really remarkable, isn't it? You're not going to miss this when you see it, green to bright blue. So this is not agar, this is a broth. But all you do is you just don't put agar into the recipe and you just leave it as a liquid, leave it as a broth. If all you're doing is looking for the results of the test, the biochemical test, you don't need agar unless you want to use the agar. You don't need the agar, you just need to know the result, right? So you can put cells into a broth, incubate them overnight, and get your result. So not a difficult test to run, the citrate test. So let's take a look at some results. For the organisms that we're using today, those 5 organisms I listed, once again, the most interesting results are between Enterobacter and E. coli. These 2 organisms can be hard to tell apart. It turns out that they also have different results on this citrate test. Over here on the left, you're looking at an uninoculated citrate plate. I used a plate here because I wanted to grow both organisms in the same container. So all I did was I put a line down the center of the plate. I smeared or I streaked some E. coli on, actually I put the E. coli over here on the right. And then I put some Enterobacter Erogenese on the left, put the plate in the incubator. 24 hours later, here we are. Not hard to read, right? It's not hard to read a citrate test. E. coli is not able to ferment citrate, but Enterobacter Erogenese can. So I'm going to get a nice, very obvious differential between those 2 organisms. Again, if I have a patient sample and I don't, if a physician suspects that one of those 2 organisms is at play in an infection, I can take those organisms, I can spread them onto citrate agar, and I can tell them apart within 24 hours. I know if it's E. coli or Enterobacter based on the citrate anyway. I would, of course, want to run additional tests to confirm which organism it was. But if the physician says, tell me if this is E. coli or Enterobacter, I can put it onto citrate and I can have a quick answer. So again, citrate test is really nice for distinguishing certain organisms from each other, including these 2. E. coli is negative, Enterobacter is positive. All right, any questions? I did want to show you with these carbohydrate fermentation tubes, and these are not inoculated, so I'm going to take my gloves off. So this is an uninoculated carbohydrate fermentation tube, and this one happens to be a commercially available one. So I just purchased this. I tell the company which carbohydrate I want in it, and they send me the media, nice and sterile, just the way I want it. I wanted to show you that the Durham tube, you can hear the Durham tube clanking around in there. You can't really see it very easily because it's full of liquid, right? It's got the media in it. But I just want to point out to you, just as a little, trick of the trade, sometimes when you buy this media, it gets jostled around a little bit. It gets jostled around a little bit while it's in shipment. And sometimes it'll arrive like this. Let me see if I can get you to see this. There's a big bubble in that Durham tube. Can you see it? There's a big bubble in the Durham tube. Compare that one to this one. So if you're going to use the media, you've got to get that bubble out. You've got to get the bubble out before you inoculate the tube. And all you have to do is just gently sort of rotate it and encourage the gas to come out of the Durham tube. All right, but it's really important that you check because you can't start off with a gas bubble. It's going to mess up your results. Remember, if you want to see a positive result, you need to check your Durham tube. And if you see a bubble, which it's hard to see, but there's a bubble in that Durham tube. There's liquid down the bottom of the tube to the left. There's an air bubble to the right. If you're trying to evaluate for that air bubble, the last thing you would want is to have an air bubble right at the start, because that could give you a false result. So just a little something to know about when you buy these kinds of test media. All right, so those are the tests that we are going to consider for this laboratory. You are now prepared to answer the laboratory questions for the week. Don't forget to get those into me by Sunday at midnight. If there are no questions about what we covered today, I'm going to let you go, okay? Enjoy the rest of your Friday and enjoy your weekend. And if you have any questions that come up before I see you again, just message me and we'll talk about them, okay? All right, everybody. Hope you have a good weekend.
Thank you. Bye.
You're very welcome.